We're continuing on with Unit 8 pre-transfusion testing. This is the first part of Part 3. It is crucial that you know the principle of every test that you do. For example, what is reacting with what, causing what type of reaction, and what do positive and negative reactions look like. We're going to discuss the following testing methods in the transfusion service: tube testing, gel, microtiter plate, and solid phase red cell adherence. Tube testing is known as the traditional method, and that's what we've been talking about up until this point in time. In tube testing, we're going to add an unknown serum, which has antibody, or an unknown cell, which has antigens, and then we mix them with a known serum or a known red cell. And agglutination indicates the presence of the antigen or the antibody, depending upon what you're testing for. For example, in the forward type, we're testing to see what antigens are present, and in the reverse type, we're testing to see what antibodies are present. Tube testing is being replaced by other techniques, but we still keep it in our toolkit because it's utilized for problem solving. Some facilities will use one system for a part of the pre-transfusion testing and then do tube testing for a different part due to the low cost. For example, ABOD typing, a lot of facilities will continue to do the tube test typing for that. There's a major hospital system here in Austin that they still use the tube system. Gel testing has become very popular, and it's based on the principle of controlled centrifugation of red cells through a dextran acrylamide gel. You have a strip or card of several microtubes of reactants that are built in, and it allows for the performance of several tests simultaneously. This can be used for detection of direct agglutination, for example, ABO, RH, red cell phenotyping, and it also can be used for the indirect antiglobulin test or the direct antiglobulin test. The red cells are allowed to interact with antibodies in the chambers at the top of the column. It is then centrifuged to force the red cells into the column to separate agglutinated from non-agglutinated cells. So once the reaction comes to completion, and centrifugation is done, the non-agglutinated cells pass freely through the gel and pellet at the bottom of the microtube. It looks like a 4+ reaction in a tube test, but it's not. Again, the agglutinated cells are too large to enter the gel matrix, and in a very, very strong reaction, they will remain agglutinated at the top because they cannot pass through the gel matrix. So in this example, you have reactions in columns A through D, and these are the varying strengths of positive reactions. The reaction in column E is negative, and you have a 4+, a 3+, a 2+, and a 1+. So here's a gel ABOD typing card. So you see that the interpretation of the anti-A would be negative, because there is no agglutination. If there was agglutination, it would be at the top because the agglutinants cannot pass through that thick gel. With anti-B, we also have a negative. With anti-D, and they use two different types of anti-D, an IgM and then a polyclonal IgM-IgG. Both of these are positive. And then the reverse type, where we have the known cells and the unknown serum. And so this patient types 4+ with the A1 cells, 4+ with the B cells. So the forward type is O+, and the reverse type is group O. In gel testing, just like in tube testing, the reactions are graded on a scale of 0 to 4+. So if you look at A, that's a 4+ reaction. All of the cells are agglutinated, and they are unable to go through the gel, so they're stuck at the top. And then we get varying strengths, where if the agglutinants are smaller, they can get through the gel a little bit, and the smaller the agglutinant, the further they can go through the gel. Look at the gel as a sieve. It will sieve, it will split out the agglutinants from the non-agglutinated. And then on the very far right in E, this is a negative reaction. All of the cells are able to travel through the gel because there are no agglutinates. One big thing to remember about gel testing, it uses microliters of plasma, very, very teeny tiny amounts, and you cannot use serum. The reason you cannot use serum is it can cause false positives if the serum is not completely clotted. And so EDTA plasma is used, and because we do not like to share in the blood bank, there are pink and there are purple EDTA tubes. And the pink is for blood bank. On the gel testing, you incubate, you spin and read. There's no washing or tube shaking. Again, it's going to go through a gel matrix to separate agglutinated from non-agglutinated cells. Sensitivity of 98% compared to the LIST tube method, so very, very sensitive. Another good thing is these cards can be saved for peer review or documented with a photo. So if your supervisor comes in on day shift or somebody with more expertise, you can actually save these gel cards versus an IAT, a Coombs test in the tube test system. You have to read those within 15 minutes. After 15 minutes, you need to start over. This system is so stable that, again, you can save it for peer review. This simplifies cross-training. It improves productivity and workflow efficiency. The only bad or downside is very expensive. Now we're going to talk about solid phase red cell adherence assays, which consists of microtiter plate hemagglutination and solid phase adherence. In microtiter plate hemagglutination, this has traditionally been used for routine processing of donor samples, but has now been adapted to automated methods in the transfusion service. It consists of a microtiter plate with 96 wells, so you can see a lot of testing can be done on a lot of different individuals at the same time. It can be adapted to either red cell testing or serum testing. You add the appropriate samples, the knowns and the unknowns, you centrifuge, dislodge the button, and it is just like the tube system except on a grander scale. So agglutination would be a positive. For example, you can see at the top right, the blue, which is anti-A, is agglutinated. The yellow, which is anti-B, is not. And then the clear is anti-D, which is agglutinated. So this individual would be A positive. Solid phase adherence has become popular because it can be automated. Red cell antigens or antibodies are immobilized attached to microplate wells to detect antibody-antigen interactions or reactions. So intact red cells or red cell membranes of a known phenotype, meaning we know the antigenic makeup of these membranes or cells. They're stuck to the side of the microplate wells. We then add our unknown, which is patient serum or plasma, which has the unknown antibodies that we may be looking for. You incubate, then you wash off the unreacted antibodies. Then, if antibody to the antigen coded on the well is present, it will attach to the antigen on the side of the well, and it sticks. How do we know this reaction has occurred? you add IgG-coated red cells, which will attach to the antibody attached to the antigen at present. So again, we have antigen, it's a sandwich technique. Then we have patient antibody to that antigen if it's present. And remember, antibody IgG has 2 fragment antigen bindings. That's 2 antigen binding sites. So when we add these antibody-coated red cells, the antibody on that red cell will attach to the antibody bound to the site of the well. So again, we have a sandwich. We have known antigens, unknown patient antibody, and then a sandwich which is really our detector to determine whether or not that reaction has occurred. You centrifuge it. If agglutinates will coat the well, and that's a positive reaction, because remember, they're stuck to the side of the well. They're not going to free flow down to the bottom. A button of cells at the bottom indicates no agglutination, which is a negative reaction. And you may want to listen to this slide or spend time with your textbook, because it's a really cool process to detect this type of antigen-antibody reaction. So slide 1 on the left shows an antibody screen and the principle. The principle is what is reacting with what to cause either a positive or negative reaction. So again, we have the red cell membrane, IgG antibody from the patient is added, and if the antigen it's directed to is present, it will stick. How do we know that occurred? Because we can't see it. We add antibody-coated red cells, and again, these are IgG-coated red cells. One of the antigen binding sites will bind to an antigen binding site on the antibody that's stuck to the well. So at the bottom you see a positive is a mat of cells, a negative is a button. On the right-hand side, we see antibody detection on the top, which we just talked about, and you can also do ABO grouping. So we can have the A antigens present in the well, then we add patient antibody that has the anti-A, and then we have group A red cells, which is our indicator system. This slide illustrates 2 panels with positive and negative reactions. Now remember, the solid buttons are negative because the antigens or antibodies are coated to the sides of the wells and will react with the patient sample if that reaction has occurred. If you look to the second row where there's a solid mat on the bottom, that is a positive reaction. So this illustrates the tube method 0 to 4+, the solid phase method 0 to 4+. Now remember, a solid button is a negative reaction in the solid phase method, and then in the gel, with the solid button at the bottom being negative and a solid button at the top being a 4+. You need to be able to interpret the reactions of these different methodologies. Automated testing platforms are becoming very popular in big donor centers or in really busy transfusion services, really large. They perform multiple tests using either solid phase, column agglutination, microtiter plate, and other types of methodologies or technologies are also being developed. Your textbook does a really good job providing an overview of these different technologies, so please spend quality time with Chapter 10. All processes from pipetting the sample to result interpretation is performed under computerized control of the automation involved. These usually interface with laboratory information systems or LIS systems, to where, just like in a chemistry department, you will be reviewing the results for accuracy. Some examples are the Galileo Echo, the Biotest Tango, and many others have come onto the market. Again, carefully review your textbook for best practices in selection of automation and the methodology that's used to perform the actual testing, the principle. The antiglobulin technique is required for testing samples from recipients for the presence of unexpected antibodies. And again, an unexpected antibody is an antibody other than anti-A or anti-B. And in selected circumstances, verifying serologic compatibility with donors. If a patient has ever had or does have an unexpected antibody, a clinically significant one, then the antiglobulin test for the cross-match must be performed. You can either use monospecific IgG or polyspecific, which has anti-C3d plus anti-IgG. Polyspecific is rarely used because what we have found out is that the anti-C3d causes false positives, and it's not really a false positive, but it picks up these nuisance antibodies. The transfusion service medical director should evaluate the time working up nuisance antibodies for determining whether polyspecific is appropriate. We quit using it years ago. It seemed like a good idea at the time, but most places now for routine testing only use monospecific anti-IgG. Let's go through the different interpretations of our cross-matches. The first is you have a negative screen and compatible cross-matches. The vast majority of the cross-matches that you will perform will have this outcome. This does not guarantee the absence of an antibody in a patient sample directed against donor antigens, or the donor cells will survive normally. Because again, remember in your immune response, once the emergency is over, antibody levels drop, and frequently with some blood group antibodies, these can fall below the level of detectability, meaning the antibody's there, but our test system isn't sensitive enough. So we go ahead and transfuse what appears to be compatible blood, and a reaction or destruction of those red cells can occur. What a negative screen simply means that there are no detectable antibodies in the patient sample directed against antigens present on these particular screen cells. And the compatible cross-match is viewed in the same manner. A patient may have an antibody, but the relevant antigen isn't present in the test system. So again, most of the time, a negative screen and compatible cross-matches will be the expected outcome for your test. So here we go. We have a negative screen. How do I know it's negative? There's zeros. There was no agglutination all the way across. Check cells, be sure you grade your check cells. A reaction of less than 2+ is not acceptable, and you'd have to repeat the screen. So again, you want a 2+ or greater, and usually 2+ is about the best you're going to get. So if there's negative reactions throughout and your check cells check, the interpretation of your screens is negative. And I put one donor. We rarely ever do a 1-unit cross-match because if a patient needs 1 unit of blood, they probably don't need any blood. Anyway, here's the example. The donor is non-reactive with the patient's serum throughout. The check cells checked. Your interpretation is compatible. It's not negative. It's compatible. The next scenario we'll discuss is when you have a positive screen or an incompatible cross-match. This could be due to either alloantibodies, the patient has an antibody against a human antigen they do not possess, or autoantibodies, which can be very nonspecific in nature and react with all cells tested. For alloantibodies, one or more screen cells are usually negative, and the donors may be compatible or incompatible depending upon whether they possess the antigen or not. If all of the tubes are positive, this could be problems with reagents, or if it's at immediate spin or 37 degrees, rouleau formation. You must identify the problem prior to transfusion. This is done by identifying the unexpected antibody and providing antigen-negative blood for transfusion. If the need for blood is urgent, then you consult with your medical director and the patient's physician. The risk of death transfusing incompatible blood may be much less than depriving a patient of oxygen carrying capacity. This is the end of Unit 8, Part 1.